is about the 2019 Nobel Prize in Medicine and Physiology, which was awarded to William Kaline, Sir Peter Ratcliffe, and Greg Semenza for determining mechanisms by which cells sense and adapt to oxygen availability. And our speaker is Peter Stacpoole, who's a professor of medicine and biochemistry and molecular biology at the University of Florida, who's very well qualified to talk in this area. Peter received his PhD in psychology from University of California, San Francisco, I think in '72.
That was in pharmacology, not psychology.
Oh, well, that's good. Thank you. That makes much more sense to me. I don't know why I found psychology. And in '76, he received an MD from Vanderbilt, and he stayed there for his residency and a fellowship in endocrinology, diabetes, and metabolism. He's an expert in mitochondrial diseases, which is appropriate since mitochondria are where oxidative phosphorylation takes place, so you can readily see the connection with today's topic. And so, Peter, we're delighted that you're here with us, and we look forward to hearing your talk.
Thank you, Ken. Thank you for the opportunity to join you in today's discussion. I appreciate the invitation. So this is as you see from the slide, can you see the first slide? Do you all see the slide?
Yes, we can see it.
Okay. So this is a rather humble attempt at alliterative humor, but what it does is to actually tie HIF with two very important elements that we'll be coming back to throughout the entire talk, and that is hypoxia and hyperplasia, and particularly neoplasia. So what we're going to talk about is not so much what I show on this slide. There are a lot of reasons, but there was recently a very extensive invited review series edited by Greg Semenza, published in the Journal of Clinical Investigation, on hypoxia inducible factors in disease, pathophysiology, and therapeutics. And I've listed a few of the focused articles on this slide. Innate immunity in liver cancer, diabetes, breast cancer, pulmonary hypertension. What links these? Well, I think what we're going to see is that there is a common theme that I hope to emphasize throughout this talk, and that is the relationship between hypoxia inducible factors and a critical energy homeostat in mitochondria. That is the pyruvate dehydrogenase complex, or PDC, and its link to kinases that regulate the activity of this enzyme called pyruvate dehydrogenase kinases, or PDKs. And we'll see how this plays out as we talk about various types of conditions associated with HIF pathology. So the outline of this talk will include a brief history about hypoxia inducible factors, how they're regulated, and then we'll focus most of the time on the pathophysiology of HIF. In that regard, again, we'll focus to some extent on the PDC-PDK axis and mitochondrial energy metabolism, and then we'll polish off with a little bit of self-aggrandizement about activities that are conducted at the University of Florida. So this is the discovery paper by, at that time, a rather young-looking Greg Semenza, published in 1992 with a postdoc by the name of Guang Wang. This was a paper that was based on their long-standing interest in studying the regulation of erythropoietin, or EPO, which is a glycoprotein that is produced primarily by the kidneys that then regulates the production of red blood cells by the bone marrow. And the red blood cells, of course, carry oxygen. And so EPO is a determinant factor in the ability of oxygen to be delivered by red blood cells to tissues. They found that EPO RNA levels were increased in response to hypoxia. They found that in cultured cells called HEP3B cells. But if any of you have ever traveled from Gainesville to go skiing in Colorado, your hypoxia inducible factor is also upregulated to compensate for the change in oxygen tension. And therefore, HIF-1 alpha was important in stimulating the production of erythropoietin in your cells so that your red blood cells could carry more oxygen and you could adapt to that elevated climb. So what Semenza and Wang discovered was what they called a nuclear factor that was induced by hypoxia and that bound to various nucleotides on the EPO gene. And that led to a transcriptional induction, which they called a hypoxia inducible factor, HIF-1, that they detected. And they said at the end of their talk, they speculated that HIF-1 might be responsible for the hypoxic activation of multiple genes. That turned out historically to be one of the great understatements of science. We now know that HIFs are responsible for transcriptional regulation of hundreds of genes. It's interesting to note that back in 1992, this was a paper, this seminal paper was published by Semenza and Wang, two authors, and was funded by a single R01 from NIDDK. A year later, they published a follow-up article in which they actually did show that HIF-1 didn't just affect the transcription of erythropoietin genes, but it also had common components that it responded to in mammalian cells in a general response to hypoxia. In other words, they began to elaborate on the number of different genes that HIF-1 might target in hypoxic states. And then 2 years later, they found that HIF-1 actually exists as a heterodimer. There's an alpha component and a beta. Both subunits are required to come together to interact with DNA. And as Ken alluded to, a somewhat older looking Semenza shared, first of all, the Lasker Prize in basic medical research with two other physician scientists. So all these were MDs, and one was William Kaelin, and the other is Sir Peter Ratliff. And subsequently, 3 years later, they received the 2019 Nobel Prize for their discoveries of how cells sense and adapt to oxygen availability. Now, I want to bring in one other Nobel Prize winner, because he and his name are going to become very important as we talk more about the metabolic consequences of HIF-1 expression, and that is Otto Warburg. He was the recipient of the 1931 Nobel Prize for his discovery of the ability of mitochondria to utilize oxygen to convert food into useful energy, which of course we know as ATP, and that this was an enzymatically driven process. Now, Warburg's name has also been lent to a process that is controlled by HIF in large part, that we call aerobic glycolysis, in which cells tend to shift from their usual way of gaining energy through oxidative phosphorylation in the mitochondria to something called glycolysis that occurs in the cytoplasm of cells, even in the presence of oxygen, hence aerobic glycolysis, which has also come to be known as the Warburg effect. And we'll see how this plays as we go forward in discussing the pathophysiology of HIF-1 alpha. Now, this is the entire HIF family that we know of so far. There are 6 isoforms. There are 3 HIFs, 1, 2, and 3, each with an alpha and beta subunit. They are sharing an N-terminus that is involved in DNA binding. They have a central region that facilitates heterodimerization of the complex. And then they have a C-terminus that recruits transcriptional co-regulatory proteins. So these are heterodimers. HIF-1 alpha is expressed ubiquitously in cells. The others, not so much. And so probably 95 to 99% of the publications you read about HIF focus on HIF-1 alpha and its metabolic consequences. Nevertheless, all of these HIFs respond exponentially to changes in oxygen over the physiological range of O2. So that begs the question, how does oxygen regulate HIF? Well, this diagram shows you two conditions. One, normoxia, normal oxygen concentration, and then hypoxia. So let's look at normoxia first. Under normoxic conditions, oxygen facilitates the hydroxylation, the addition of O2, OH groups to the HIF-1 alpha protein. That also facilitates the binding of another protein complex called the von Hippel-Lindau complex. The combination of that signals the ubiquitination and ultimately proteasomal degradation of HIF-1 alpha. So, normal oxygen tension, low HIF-1 alpha. What about when oxygen is in short supply? Then there's no block to hydroxylation or to von Hippel-Lindau protein aggregation. And so HIF-1 in the cytoplasm translocates to the nucleus to join its HIF-1 beta partner. And together, they facilitate transcription by binding to hypoxia response elements, shown here as HRE, and activate transcription of whatever genes are appropriate. Now, as I said before, HIF has been associated with the transcriptional control of hundreds of genes. And this is just a snapshot of some of the ones, because it's these that in many ways become relevant to our discussion later on of pathology. Glucose and energy metabolism, many enzymes of glycolysis of the glycolytic pathway, and also an enzyme here that I will continue to emphasize, and that is pyruvate dehydrogenase kinase. This is a mitochondrial enzyme, but as we'll see, it plays an important role in terms of the metabolic effects and consequences of HIF-1 alpha overexpression. Genomic instability, angiogenesis, metastasis, epithelial mesenchymal transition, regulation of pH, cell proliferation, invasion. There's a common theme here, and that common theme has to reflects the effect of HIF-1 alpha in regulating processes associated with neoplasia, both benign and malignant, as we'll see. Well, you can imagine that once Semenza and Wang published their early findings, things began to percolate in terms of the HIF-1 field. This just shows you a brief snapshot of about a little over a decade of the response to the interest in the scientific community of HIF-1. And it shows really on the upper right-hand side the number of publications from 2000 to 2012. Well, as of last month, I calculated through PubMed over 22,630 citations. Publications, excuse me. The number of citations has increased appropriately. And then if you look at the lower left-hand corner of the slide, you see in the orange bars, this waxing and waning of clinical trials. So once people became more aware of the consequences of HIF-1, one then thought that one could target that therapeutically and regulate HIF-1 and perhaps be a target for the treatment of cancer or other types of diseases. Well, that hasn't developed too far, but I'll mention in just a minute a few things that have led to clinical trials. What I want to do is remind you about what aerobic glycolysis or Warburg metabolism is. This is a picture of glucose on the outside of the plasma membrane of cells, transported by glucose transporters, and then undergoes a series of enzymatic reactions called glycolysis that lead to pyruvate. Pyruvate has a number of fates, but one of the fates, and the predominant fate under normoxic conditions, is to be transported from the cytoplasm into the mitochondria. And once inside the mitochondria, it is irreversibly decarboxylated or broken down by the pyruvate dehydrogenase complex to convert it to acetyl-CoA. Now, acetyl-CoA has innumerable important effects in intermediary metabolism. But what's shown here is that a major consequence of acetyl-CoA formation. In other words, the formation of these glucose carbons is to enter what's called the tricarboxylic acid or Krebs cycle, or citric acid cycle. And by doing so, glucose either biosynthesized by the liver or taken in the diet by carbohydrates, complex carbohydrates, can then enter the TCA cycle, tricarboxylic acid cycle, and be further metabolized. Now, the processes of glycolysis and TCA cycle activity have a number of other consequences. One of them, and that's important in terms of considering neoplastic conditions, is that some of this glucose carbon gets shunted into a pathway called the pentose phosphate. That's a misspelling. That should be phosphate pathway. That provides not only the generation of glutathione and NADPH for antioxidant protection of cells, but also nucleotide carbon for new DNA and RNA. In addition, the TCA cycle can also generate molecules involved in the synthesis of fats and amino acids, the building blocks of proteins, and also provides building blocks for nucleotide synthesis. So all these factors can be involved and can be regulated to increase or decrease the substrates for new cell growth, new cell formation. What you see in red here are some of the enzymes that HIF-1 alpha increases expression of. It increases the uptake of glucose and glycolysis, but it also converts pyruvate not to enter the mitochondria, but to be reduced in the cytoplasm to lactate and accompanying proton, so lactic acid, which can then exit the cell through monocarboxylate transporters that are also upregulated by HIF. HIF also increases the activity and the expression of pyruvate dehydrogenase kinases. And this interaction is important, and we'll talk about this in more detail. But I just wanted to mention in passing that this is a partial list of the growing list of potential HIF-1 inhibitors that are designed potentially as therapeutic agents for the treatment of neoplastic conditions, particularly cancer. And you can see they, depending upon the molecule, they interact at different levels of HIF metabolism. To the best of my knowledge, a few of the HIF inhibitors have entered into early phase clinical trials. None have progressed beyond that. And you might wonder whether or not targeting a protein like HIF that has so many protein effects on cell metabolism and cell growth and health is particularly wise, because if you do have a non-selective inhibitor of HIF-1 or HIF-1 alpha, you can imagine other things that can occur that might be deleterious to the host. Nevertheless, that is an area of active research. So before we go further into understanding how and where HIF works in metabolism, in general, the evidence indicates that there is a general host response to hypoxia that leads to cell proliferation and to this shift of to favor glycolysis over oxidative phosphorylation by the mitochondria. This tends to limit oxygen consumption, but it also tends to limit production of reactive oxygen species, abbreviated as ROS, that can provide oxidative stress and damage to cells. And the seat of the soul for reactive oxygen species generation is the mitochondria. So by suppressing the mitochondria, we also suppress reactive oxygen species production. There's also, as a general response to hypoxia, an angiogenic, a increase in the transcription of angiogenic factors, factors that induce new blood vessel growth. Now that is ostensibly to provide increased oxygen availability, and indeed in wound healing, which is also governed in part by HIF overexpression, that is a physiological and advantageous response. But in tumors, the increase in angiogenesis is often disorganized and tortuous, and it really doesn't increase oxygen availability, certainly to many parts of the tumor microenvironment. Nevertheless, that is a consequence of HIF overexpression. So this has led to sort of a general postulate that neoplasia, whether it's benign or malignant, has a propensity to establish and to perpetuate a hypoxic microenvironment. And this may be a primary mechanism that underlies this pathology, being the stable upregulation of HIF-1 alpha. And HIF-1 alpha does what it's supposed to do. It promotes cell maintenance and growth by favoring glycolysis over oxidative phosphorylation, angiogenesis over vascular stasis, and survival over apoptosis. And apoptosis, which is programmed cell death, is a natural process to get rid of damaged cells. And many of the factors that are elaborated by cells to induce apoptosis are elaborated by mitochondrial proteins, such as involved in the respiratory chain and in caspases. So many physiological and pathological processes may exhibit the biochemical hallmarks of neoplastic disease. And so this is a, I hope not too off-putting slide that summarizes further the basic elements of how we convert the foods that we eat and the substrates that we biosynthesize in our bodies to useful energy in the form of ATP. So glycolysis, as I mentioned before, occurs in the cytoplasm, leading to pyruvate. The pyruvate has more than one fate in the mitochondria, but under normal conditions, most of it is shunted through the pyruvate dehydrogenase complex to acetyl-CoA, shown here. So does the breakdown of long-chain fatty acids, the fats that we absorb or biosynthesize, through a process called beta oxidation. That also yields acetyl-CoA, which enters the tricarboxylic acid cycle. Now, enzymes from beta oxidation, the pyruvate dehydrogenase complex, and several enzymes of the TCA cycle generate what we call reducing equivalents. They are molecules abbreviated as NADH and FADH2, and they provide electrons to the first complexes of what's called the respiratory chain. The first four complexes of the respiratory chain utilize these electrons and ultimately detoxify the oxygen we breathe and convert it to water. The final or fifth complex of the respiratory chain utilizes a proton gradient that is created by its predecessors by converting inorganic phosphate and ADP to the principal energy substrate ATP. Mitochondria make about 95% of our ATP. So it is the often called the energy powerhouse of the cell, and well named. Now, again, I know this is getting a little deep in the woods, but I wanted to emphasize the importance of the pyruvate dehydrogenase complex, because this is perhaps, one could argue, the most important energy homeostat in our bodies. It takes pyruvate. The complex, by the way, is not just one enzyme. It's a collection of enzymes bound together to produce a final product, and that is acetyl-CoA and NADH. So it takes pyruvate, and the first part of the complex, which is called E1, which is a heterotetramer of 2 alpha and 2 beta subunits, decarboxylates pyruvate, and in the presence of thiamine pyrophosphate, thiamine being a B1 vitamin, ultimately allows the production by the E2 component of acetyl-CoA. And through redox mechanisms, the third component generates from oxidized NAD plus, to NADH. And so the net reaction is pyruvate plus NAD plus coenzyme A, shown here, to form acetyl-CoA, NADH, and carbon dioxide. This is probably the largest enzyme complex in our bodies. The molecular mass is around 9.5 to 10 million Daltons. That depends upon species, but in humans, that's what it seems to be. And you can see a schematic diagram of what it appears to look like, and that is the outer core is the E1, that's the decarboxylating enzyme, And that's bound and stabilized by the E2 and E3. Now, this is a prototypical structure that's also shared by two other very important energy metabolizing enzymes. And one is the oxoglutarate dehydrogenase, which is part of the TCA cycle, and then also the oxoacid dehydrogenase that metabolizes branched chain amino acids. Now, over the last very few years, a remarkable discovery has been made, and that is that this massive complex is translocated intact and functional from the mitochondria to the nucleus. How it does that is still enigmatic. Nevertheless, it is thought that the nuclear PDC may be important in generating acetyl groups for histone acetylation and therefore epigenetic regulation. Now, like many important regulatory enzymes, it itself, the PDC itself, is regulated very tightly. And the most well-studied and perhaps the most physiologically important process of regulation is through reversible phosphorylation. When this reaction occurs, PDC must be unphosphorylated. It must not have any phosphate groups attached to certain serine residues of the E1 alpha. If it does, those are facilitated by pyruvate dehydrogenase kinases that phosphorylate the enzyme and render it catalytically inactive. There are also pyruvate dehydrogenase phosphatases that remove the phosphate and restore enzyme activity. You can imagine that this knowledge has then beget ideas about targeting this for therapeutic purposes. And the prototypic inhibitor of pyruvate dehydrogenase kinases, thereby blocking PDC in its unphosphorylated active form, is a very simple molecule called dichloroacetate, or DCA. It's just vinegar with a couple of environmental pollutants attached to it. But it's a small molecule and small enough, and it looks enough like pyruvate to be able to bind to the binding pocket for pyruvate on PDKs. Pyruvate, being a substrate for the enzyme, locks onto PDKs and inhibits the phosphorylation, keeping the enzyme, the PDC enzyme, active so pyruvate can be metabolized. DCA fits into that pocket itself. Now, the studies of DCA over the years have generated a very rapidly growing cottage industry from both academia and pharma in the generation of other potentially patentable PDK inhibitors. DCA being so small, common, and long-standing is not patentable. And these new drugs either have DCA molecules attached to them, or else they're too large to fit in here and don't have DCA. So they attach to other binding pockets on the PDK and regulate its activity. Again, stimulating PDC activity in the process. So now I'd like to turn to pathology associated with HIF1 upregulation, and also in relation to regulation of the pyruvate dehydrogenase complex, because HIF1, as we saw earlier, is a regulator and increases the expression of the kinases that inactivate this enzyme. Now, when we think of metabolic flexibility, we think of the ability by cells to switch between metabolic pathways in order to maintain their normal energy status and homeostasis and to conduct their normal physiological functions. But the loss of metabolic flexibility underlies the pathobiology of innumerable types of diseases that on the surface don't seem to be connected, but from a molecular and biochemical standpoint, they definitely are. Loss of metabolic flexibility at the level of this axis is associated with many conditions, some of them physiological, some of them pathological. Aging, insulin resistance, obesity and diabetes, inflammatory diseases, cardiac ischemia and failure, and neoplastic conditions, both physiological, such as the differentiation of the conceptus, the pre-implantation conceptus, the activation of immune cells, and the process of wound healing are all physiological neoplastic conditions that are associated, in some cases, with upregulation of HIF1. But there are non-physiologic or malignant neoplasias, including cancer, and two other important conditions that I'll touch on briefly that are not malignant, but are nevertheless neoplastic conditions associated with pathological upregulation of HIF1, and that is pulmonary arterial hypertension, or PAH, and restenosis of coronary arteries. So let's first turn to the relationship between HIF1 alpha and obesity. And this was first studied in animals fed either a high-fat diet or in mice who were genetically predisposed to obesity. And here they could measure tissue hypoxia. And although this slide doesn't reproduce it very well, and you may not be able to see it, what happens on a high-fat diet is that these adipocytes, these adipose cells, undergo both hypertrophy, meaning expansion of the cell itself, and hyperplasia, meaning an increase in the number of cells. So this is a neoplastic condition, albeit non-malignant. As a result, there is outstripping by the cells of their vascular supply, so they perpetuate a hypoxic environment. When you look at the level of hypoxia in muscle and white adipose tissue, since this is an animal that is predisposed to adipose increased mass, that you can see that hypoxia really is isolated to white adipose tissue. Now, as a measure of Warburg metabolism or aerobic glycolysis, remember when upregulation of HIF1 alpha shunts pyruvate away from mitochondria and into lactate formation, lactate production in these hypoxic adipocytes increase. So oxygen supply can't meet the demand of expanding adipose tissue, resulting in relative hypoxia and increased lactate production, which is a reflection of Warburg metabolism. And if you take mice that are genetically predisposed to obesity, these classical OB mouse, they exhibit induction of HIF1 alpha that is also selective to white adipose tissue. And what you see here is that not only is the hypoxia genes associated with obesity, including HIF1 alpha itself in terms of its increased expression, but also the glucose transporter that allows glucose to enter the cells, and also upregulation of the pyruvate dehydrogenase kinase 1 that inhibits PDC. In muscle, there's no change. It's isolated to adipose tissue. The same for mice on a high-fat diet. Upregulation of HIF1 in fat, but not in muscle. Selective induction of Warburg metabolism. Hypoxia in vivo can recapitulate this type of phenotype. And without going into any of the detail here, let me just focus on this, this color slide or figure that indicates that when fat expansion occurs and hypoxia occurs, HIF1 alpha is upregulated. It induces genes associated with tissue fibrosis and oxidative stress. And this leads to fibrotic changes and inflammatory changes in the target tissue that lead to insulin resistance. Now, what happens if you do the opposite, if you block HIF1 alpha? Well, that decreases adiposity and reverses some of the metabolic changes that we saw earlier. If you knock out HIF1 alpha genetically, you decrease fat formation, you increase resistance to dietary-induced obesity without a change in food intake. So here's an example whereby body mass actually falls a little bit with a knockout, but the fat mass drops precipitously. And knockout animals of HIF1 alpha show a marked decrease in the size and, although not well shown here, the number of enlarged adipocytes. And as a consequence of that, they improve their insulin sensitivity, decreasing insulin resistance. And this can be seen here in measuring the blood glucose excursion in animals that are control, fat animals, and those who have HIF1 knockout. So with any glucose challenge, there's less of a rise in blood sugar, and it reaches lower levels sooner than animals in whom HIF-1 alpha is hyperexpressed. The same thing occurs here. Less insulin is needed in the knockout animals to achieve this level of glucose tolerance. And these other parts of the slide show the same general message, and that is, there's an example of where you can lose weight without dieting. Just knock out your HIF-1 alpha and you're fine. This occurs in humans. This is a study in which visceral white adipose tissue of obese humans and mice were studied. It showed that HIF-1 inhibits the oxidation of fatty acids and therefore maintains fat stores. When you biopsy the white adipose tissue of individuals, you can see that not only are these people have greater body mass, but also their fasting blood sugars are higher, their 2-hour blood sugars following a glucose meal are higher. They also have changes in certain genes that promote inhibition of fatty acid oxidation and promotion of fat storage. And you can see that in these obese individuals, the expression of HIF-1 alpha is markedly increased compared to lean counterparts. Now, what happens if you induce weight loss? And in a study of morbidly obese patients before and 3 months after bariatric surgery, there was a striking change in both the histology of adipose tissues with a reduction in the number and size of adipose tissues, shown both histologically here and by bar graphs here, but also a change in the expression of HIF-1 alpha. Here's a control. These are prior to surgery, and this is 3 months following weight loss reduction surgery. In addition, there were improvements in insulin and other acute phase proteins are associated with insulin resistance and inflammation. So histological, metabolic, and inflammatory changes are potentially reversible with weight loss in humans, and much of this is mediated by changes in the expression and the metabolic consequences of HIF-1 alpha. Let's turn to another disease in relation to HIF-1 alpha, and that is cancer. Now, again, a central problem in this is upregulation of pyruvate dehydrogenase kinases and downregulation of the mitochondrial enzyme pyruvate dehydrogenase complex. HIF, as well as other oncogenes such as Wnt and Myc, can all stimulate the upregulation of pyruvate dehydrogenase kinase. And what does it do? It phosphorylates serine residues on the E1 alpha subunit of PDC, blocking its activity. The consequence of that is to decrease oxidative phosphorylation in the mitochondria, reduce the production of reactive oxygen species, and also inhibit the elaboration of cytochrome C and caspases that normally mediate apoptotic cell death. In addition, upregulation of PDK increases through aerobic Warburg metabolism lactate production. Lactate, as well as pyruvate and protons, create a positive feedback loop because they tend to stabilize HIF-1 alpha and retard its degradation, leading to more activation of PDK, inhibition of PDC, and so this positive feedback loop is part of the pathology. Lactate is a highly bioactive molecule. It's not a waste product of glycolysis. It is directly involved in facilitating immune suppression, in stimulating angiogenesis, and in promoting metastasis in many studies of experimental tumors. Combined, the increase in Warburg metabolism and the decrease in oxidative phosphorylation increase biomass, facilitating new cancer cell growth, and lead to an anti-apoptotic pro-proliferative condition that preserves survival in tumor cells. Now, that begs the question whether or not you can therapeutically target PDK and achieve some benefit in patients with cancer. And there are a few phase 1 trials, one of which was conducted at the University of Florida, that have looked at chronic administration of the prototype inhibitor dichloroacetate, DCA, in patients with recurrent brain cancers, as well as other solid tumors. Really, this is on the background of well over 100 and continuing to accelerate number of preclinical studies looking at both solid tumors and hematologic malignancies in a way in which targeting the PDK can have potential therapeutic benefit and also reduce tumor resistance to standard chemotherapy agents as well as exoradiation. So in a study of 5 patients with recurrent glioblastoma, which is the most common primary brain cancer in humans and also almost a death warrant because very few people survive more than about a year or a year and a half after diagnosis. It was found that examining the biopsy tissue of these patients, there was an upregulation of the second isoform, PDK2, of the pyruvate dehydrogenase kinase. In addition, not only was PDK2 upregulated, but when these tumor cells were subjected to DCA ex vivo, HIF1 alpha expression fell, PDK2 expression fell, as we would expect. There was an increase in oxidative metabolism as measured by an increase in ROS production, the active oxygen species production. The mitochondria were depolarized, angiogenesis decreased, and cell apoptosis also was stimulated. When DCA was administered for up to 15 months to these patients, there was positron PET evidence, scanning evidence of a stabilization or a decrease in glioblastoma in vivo in some of these patients, providing proof of principle that targeting the pyruvate dehydrogenase complex, pyruvate dehydrogenase kinase axis could have therapeutic benefit in tumors, especially those in which HIF1 alpha predominates. So let's turn to another neoplastic but non-malignant condition, pulmonary arterial hypertension. This is a condition in which the biochemistry is very similar to what we just saw in cancer. It is a fatal disease, ultimately, because it leads to neoplastic proliferative narrowing of the vascular lumen by pulmonary vascular endothelial and smooth muscle cells that cause narrowing of the lumen, strain on the right ventricle, and patients typically die from right heart failure. Now, there are FDA-approved therapies, but they are relatively modest in their efficacy. Many preclinical studies have shown that when you induce pulmonary arterial hypertension in a rodent model, such as by subjecting them to chronic hypoxia, shown here as CH, the lumen of these vascular cells, compared to normal, is virtually obliterated. However, if you treat these cells by the PDK inhibitor DCA, these hypoxic cells reopen their lumen because apoptosis occurs and proliferation decreases. So this has led to so far, one phase 1 trial that was conducted both in Canada and in the UK in adult patients who had pulmonary arterial hypertension. And what they showed was that small doses of DCA administered over the course of about 4 months decreased mean pulmonary arterial pressure, which is abnormally high when you consider the constriction of the vessels. It also reduced peripheral vascular resistance. And that's important too, because vascular resistance increases as a result of the proliferative changes. And in terms of function, patients were able to walk further during a 6-minute walk test, which is a standard measure of functionality in such a disease, when they received DCA. So 6-minute walk test increased. All these changes were modestly significant. And so this is going to be leading to what we think will be a phase 2 trial, multi-center phase 2 trial in the not too distant future. How about coronary restenosis? Another apparently diverse disease When patients have stents, they will sometimes result in stent failure. And the mechanism by which stent failure occurs is often the neoplastic proliferation of coronary endothelial cells and smooth muscle cells, very similar to pulmonary arterial hypertension. And the basic biochemistry, again, mirrors that we showed in cancer. So it's been found in a study from, I believe, France, that pharmacological or genetic inhibition of PDK decreases myointimal formation in vitro and in vivo. Here's an example of using DCA in human coronary arteries, showing the change in luminal size, in rat aorta, in the rabbit iliac artery, which is an offshoot of the abdominal aorta, and following genetic knockdown of PDK2 in human coronary arteries. Again, showing that modulation of the PDC-PDK axis might have therapeutic potential in terms of reversing the hyperproliferative state in coronary restenosis. How can HIF-1 alpha affect sepsis? You know, sepsis and septic shock represent the most common cause of in-hospital mortality in the developed world. Patients have about 25 to 30% of patients will die in the hospital from septic shock. And if you follow these individuals for 3 months, up to 40 to 50% will die because their essential organs, whether it be liver, heart, lung, brain, etc., fail to recover and ultimately die in part from energy failure. Since I was in medical school, and when Ken was in medical school, the treatment of sepsis and septic shock hasn't changed. The basic 3 factors that are involved in treating patients with septic shock are giving them fluids to increase their intravascular blood volume, giving them antibiotics to try to treat the infection, and giving them drugs that increase the pumping function of the heart called pressors, such as norepinephrine, an adrenal medullary hormone. This is an inflammatory process, and these cytokines that are elaborated in these conditions stabilize HIF-1 alpha, even under normoxic conditions, and lead to Warburg metabolism and consequent perturbation of the PDC-PDK axis. And so we collaboratively worked with investigators from Wake Forest to recapitulate a classical model of urine sepsis, and that is to perform an abdominal incision, remove the cecum, part of the large intestine, tie it off, isolate it from the rest of the intestine, and then puncture it, replace it back into the abdominal cavity, sew it up, and that precipitates a severe bacterial peritonitis and systemic sepsis. What we found was that the phosphorylation of PDC-E1 alpha was markedly increased in control animals, but was dramatically decreased with the addition of DCA. The total amount of the enzyme did not change, but it was the phosphorylation state that was regulated here, and that's shown here graphically. The consequence of that is multifactorial. For one, mean arterial pressure increased with activation of PDC. In animals with who have had this cecal ligation and puncture, if you look at the histological profile of the intestine, it looks like a grenade went off inside. The architecture is completely destroyed. But when you give animals a PDK inhibitor like DCA, architecture is restored. This is associated with the expression of beta-catenin, which is an anabolic transcription factor associated with this type of phenomenon. In addition, and as a consequence, not only is end organ reversal occur, and this has been subsequently confirmed in other studies that we've done, but survival is markedly increased in animals in which the PDC is reactivated. And that also is associated with an increase, a significant increase in the bacterial clearance by the host of the bacterial that was produced by cecal ligation and puncture. So I just want to summarize by saying that there are many superficially disparate conditions that share common pathology. Hypoxic and proliferative diseases share many aspects of Warburg metabolism and induced by overexpression of HIF-1 alpha. It's interesting that some of these are also expressed in the cells of children who are born with loss of function mutations in the PDC, leading to congenital lactic acidosis as one of the life-threatening complications of congenital pyruvate dehydrogenase complex deficiency. These children suffer and die from progressive neurological and neuromuscular degeneration because of mitochondrial energy failure. So the question is whether or not the ability to stimulate whatever residual normal enzyme activity exists in these children could be therapeutic. And so that brings us to the final slide, and that is the relationship between everything I've said and UF. There are a number of translational research studies and clinical research studies that are either active or are being pursued at the University of Florida. We're running a multi-center, federally funded, pivotal phase 3 trial of oral DCA in children who have congenital PDC deficiency. But obviously, in order to be able to be born and to live, they must have some normal residual activity. And the hypothesis is that by dephosphorylating and activating residual normal enzyme activity, we may be able to improve their morbidity. We're not looking at changes in mortality in this particular trial, but the hope is that by improving their energy metabolism in cells, this will benefit them clinically. There's also ongoing work in understanding the developmental pharmacology of this drug and how it may translate into other clinical activities. There's a project that's ongoing now, federally funded, in which pregnant sheep are studied as a model for life-threatening complications during parturition in humans. In difficult deliveries, hypoxia occurs and may cause potentially fatal cardiac arrhythmias and failure in the offspring. We found that DCA in pregnant sheep can cross the placental barrier and be pharmacologically active in the fetus. And so what we're trying to do now is to determine whether or not under stress conditions, the drug may decrease HIF-1 alpha expression and PDK expression, which is upregulated in this stressful condition, and potentially lead to a new avenue for therapy for these critically ill newborns. The National Institute of Aging has accepted the proposal, and they are going to be studying in inbred strains of mice the effect of lifelong administration on DCA on both health span and lifespan. There is a pending application to conduct a multi-center phase 2 trial in glioblastoma patients. There is a phase 2 trial undergoing early development for a multi-center trial of DCA and pulmonary arterial hypertension, and also a pending multi-center trial in septic shock. And finally, just to cap it off, because DCA rapidly crosses the blood-brain barrier and does the same thing in brain cells as it does in peripheral tissues, modulating energy metabolism in the brain and lactate metabolism, colleagues and I have hypothesized that the effects of DCA may limit the desire of recipients of drugs of abuse to continue taking those drugs. And there is a standard validated animal model in which rats self-administer cocaine, in which we will be studying the effects of DCA to see whether or not that behavior can be ameliorated. all of these conditions, all of these apparently disparate diseases target a single axis, PDC, PDK axis. And with that, I thank you, and I'm happy to take questions and comments.
Thank you. Ken or John, would you like to comment before we go to questions?
John?
Ken, what ramifications of this discovery is enormous, and I can understand that there would very likely be, can we get a view of everybody? Anyway, likely be unintended side effects because affect one thing and it could have effect on, you suggested that before, which makes it a little risky, I would think.
That's a very logical question, and one that has been addressed. We know, the question really is chronic activation of the pyruvate dehydrogenase complex good for you? And the only human evidence we have that closely relates to that is that children with congenital forms of lactic acidosis have now been treated with DCA chronically for a dozen or more years. In fact, three children have been studied for over 25 years with chronic oral administration of the drug, with no change in any parameter associated with hematological function, liver function, kidney, et cetera, et cetera. And I think that's probably the best evidence. These are very vulnerable children, obviously. They have limited capacity, both intellectually as well as in terms of immunological function, et cetera. And the drug seems to be well tolerated in them. The one clinically limiting toxicity of DCA that has been found mostly in adults, but much less so in children, is a reversible peripheral neuropathy. And we don't know the mechanism for that at this point. But what we have done is to identify genetic polymorphisms in the gene that encodes the drug metabolizing protein for DCA, which is the only protein, which is the only drug that's metabolized by this, which is the glutathione transferase. And we can dichotomize patients into fast metabolizers of the drug and slow metabolizers. And so we dose people accordingly. And by doing that, we seem to, if not eliminate, then markedly reduce potential for toxicity.
When you mentioned that these children have other aspects of their disease, if one could identify this disease in utero or something like that, could one avoid those consequences?
It's been attempted and unsuccessfully. Now, modern genetic testing, prenatal testing, might provide a potential. One of the issues is that about 80% or more of the children have a mutation in the E1 alpha subunit of PDC. That's the one that decarboxylates pyruvate to CO2. That enzyme is X-linked. And so males tend to have a very severe course. Females, it's highly variable. We've studied one pair of identical twins, and at presentation, one child had severe lactic acidosis and very severe neuromuscular degeneration and neurological degeneration. The other one had a much more benign course, and she is one of the children who has survived longer than anybody else.
How common is this condition?
It's a rare disease, obviously, and it's identified by the FDA as a rare disease. We think that the prevalence of PDC deficiency in this country is probably on the order of around 250 to 300 patients.
Yeah.
But it's not a rare disease if you have it.
Yeah, I understand.
Peter, you talked about, and I don't remember if it was preclinical or clinical, although it could well be clinical, your studies with septic shock.
Preclinical.
So, microbiologist, I wonder if you could just elaborate on that a little bit.
Sure. Well, sepsis was traditionally viewed as predominantly an immunological problem with elaboration of cytokines that caused damage to cells. That stimulated over 50 clinical trials, all of which failed, that focused mainly on trying to address the immunological changes that occur in sepsis and septic shock. Millions of dollars failure by pharma. Over the last probably less than 10 years, I would guess, there's been a shift in understanding that this is an immunometabolic problem, and that metabolism of inflammatory cells, the innate immune cells, and the consequences of principally bacterial sepsis can modulate metabolism and in turn, therefore, lead to changes in immune function. What we found in not only the studies that I summarized, but also in subsequent studies in vitro and in vivo, is that DCA, by stimulating PDC, seems to decrease inflammatory cytokine production and modulate both the metabolic and immune function of cells in response to sepsis, and preserve end organ function. And we've seen this in the heart, in the liver, and in other end organs, including the gut, which I showed a picture of. And to us, it's still a remarkable phenomenon that still remains to be fully worked out, is that by changing metabolism, you change immunological function in response to an invading organism.
Thank you. We have a question over here. Alan March.
Alan?
Yes, I have a question. In patients who have the COVID-19, who suffer a cytokine storm and an excessive oxidative stress, is there an upregulation of the HIF-1 alpha, or is that postulated? And how is that related?
I've tried to keep up with that literature, and I've never seen an article that focuses on HIF-1 alpha in COVID-19. Maybe someone else in the audience has, but I've not seen it.
There's an opportunity, Peter.
Yeah, right. Well, we tried to interest NIAID in considering a trial of DCA in COVID-19 patients, actually. We thought it would be safe and potentially effective, but we couldn't get the way in which NIAID responded to COVID was to establish a hierarchy of types of grants to which you could apply, and none of them were ones that we could use and we could study. We even contacted Tony Fauci early on because one of my collaborators knows him, and he was gracious enough to reply, but didn't go anywhere.
I think he has his mind made up early on.
Yeah.
Where he was going. I thought Roy, as a noted oncologist, might want to comment on the consequences for tumors.
Well, hello, Roy. It's been a long time.
You're muted, Roy.
I think you've described well some of the molecular aberrations that occur in cancer cells. The linkage that still is puzzling is how the molecular aberrations lead to proliferation of viable cells, even though the cells are cancerous and don't behave physiologically as normal.
Yeah, but the basic biochemical structure of those cells is no different from somatic cells, and HIF works in the same way as it does in promoting proliferative cells in adipose sites, as it does in cancer cells. The basic mechanisms are by stimulating Warburg metabolism, the offshoots of that lead to a decrease in oxidative phosphorylation, an increase in building blocks for new cell growth, and an increase in antioxidants that help preserve and defend against oxidative stress by these tumor cells. So I think that the proliferative mechanism is not unlike what you see in non-malignant proliferative diseases, whether it's pulmonary arterial hypertension or obesity.
So the oncogenic process occurs independently.
Yeah, well, yeah, I think it probably does. And in those cases, there are particular oncogenic mechanisms that induce upregulation of HIF and upregulation of PDK. So yes, sure. The inciting agent is different, but the consequences are very similar.
Okay, thank you.
Now, as our neurosurgery group studying glioblastoma in clinical trials.
Yeah, they're doing that. We're doing that. We're doing our work independent of them. We're looking at, right now, we're looking at cell cultures of human glioblastoma to try to determine which cells might be the better targets for DCA, and then to try to translate that into humans. And so we've collaborated with what is now called the NCI-sponsored Adult Brain Tumor Consortium that is headquartered at Johns Hopkins and involves a number of cancer centers around the country. And if our proposal is accepted, we'll conduct a multi-center trial using those clinical sites at Hopkins and Harvard and Texas, et cetera, to study the effects of DCA in a trial in which we will treat half the patients before their debulking surgery. All these patients require at least one debulking surgery. We'll treat the half of the group for a week with DCA and the other half not. Then when they go to surgery, we will take out that tumor tissue, and we, in our laboratory, will look at various tumor markers, including whether or not the phosphorylation state of PDC has been changed. And then after that, after the debulking surgery, all patients will receive open label DCA, and we'll look at outcome.
Well, if there are no other questions, is there a question over here?
I don't see any others.
Okay. Well, in that case, Peter, thank you very much. You're very gracious. Peter was scheduled to talk in